Welcome to this lecture series on eukaryotic gene expression, basics and benefits. Today we are going to discuss about a very fascinating area of biology and medicine known as the human gene therapy. The last two classes we spent considerable amount of time discussing about introducing genes into mammalian cells using viral and non-viral vectors. This is what is shown here. We spent considerable amount of time discussing how we can introduce genes and express proteins in mammalian cells using non-viral vectors as well as viral vectors. Now, once the technology for introducing genes into mammalian cells and expressing them successfully using viral or non-viral vectors were developed, then people started asking the question, instead of introducing genes into mammalian cells, expressing their proteins and then purifying the proteins and then using for therapeutic purposes, we are going to use these non-viral and viral vectors and directly introduce these vectors into the humans itself, so that these vectors would go to specific tissues, transfer specific cells, and the gene will be expressed and protein will be made right inside our body. So you don't have to express your genes either in using yeast expression system or E. coli expression systems or insect or mammalian expression systems, then purify the protein and then give it as a therapeutic. Instead, use these mammalian expression systems, either viral or non-viral, directly inject these vectors into the humans and see whether the protein can be made right inside our body. This methodology of using viral or non-viral vectors to introduce genes into the body with the objective of curing a disease has now come to be known as gene therapy. It's a very exciting area of research. So today we will focus on human gene therapy. So the question we are asking is, can the non-viral and viral vectors, which we have been discussing about using and expressing genes in mammalian cells in culture, can there be vectors encoding that particular gene directly into the human so that the therapeutic protein will be made inside the humans and the disease can be cured? So, can the non-viral and viral vectors be directly introduced into tissues of animals including humans to cure a disease? The answer is yes. So, let us now see how we can do this. So, the definition of gene therapy basically is the treatment of genetic or acquired disorders by delivering normal genes to correct that particular disease. This is what is the very simple definition of gene therapy. The gene therapy can be used for correcting defective genes that are responsible for disease development. There is many ways of doing this. In the normal way, usually most of the gene therapy protocols, you know that there is a genetic defect, there is a gene which is mutated or it is deleted, and as a result a specific protein is not made. What you now do is, you take the correct gene or the normal gene for that particular protein, put them in a viral vector, non-viral vector and introduce this into the human. So this is where you are basically supplementing the normal gene to the human beings, but the defective gene is still there. But the function is restored by expressing the normal gene, so the therapeutic protein will be made. So, a normal gene is expression basically a supplementing the individual with a normal gene so that the normal protein can be made. This is most of the gene therapy protocols is aimed at this particular kind of a approach. But a much more ideal approach would have been to actually exchange or replace the defective gene with a normal gene by actually what is called a homologous recombination. So you can actually replace the defective gene which is present in the chromosome with a normal gene so that the defective chromosomal copy of the gene is removed and the normal gene is expressed. So this is the ultimate object of gene therapy, but we still have not reached this level of sophistication to aim at gene replacement therapy. The other thing that people are trying out is what is called as suppress the expression of a defective gene. Usually in diseases like cancer, when you know the cancer is because of expression of oncogenes, overexpression of oncogenes, using molecules such as antisense or RNA or what are called as siRNAs or miRNAs, which we will discuss in much later classes, can you actually block the expression of the endogenous gene. So this is called a gene suppression. A much more challenging aspect of gene therapy is what is called as a repair, in vivo repair, wherein an abnormal gene, if a particular gene has a mutation or a deletion, can you repair this endogenous gene by actually introducing the correct gene so that the endogenous homologous recombination or a gene repair DNA repair pathway now through homologous recombination replaces the endogenous gene with the respective gene. It's called as a endogenous repair. So, number of approaches are possible when we think about introducing a gene to correct a particular genetic disorder, but the most common approach that is being practiced or being tried out in various clinical trials is to see if a defect is there in a particular gene and as a result a specific protein is not being made. Into such individuals, can we introduce a normal gene through a non-viral vector or viral vector so that that particular protein can be made and the patients can be cured of a particular disease. This is what we are going to discuss now. The gene therapy is kind of a extension of a protein therapy which we have been discussing, because the last about 5 to 10 classes we have basically been talking about various expression systems, how we are using E. coli or yeast or insect or mammalian expression systems to make a number of proteins of therapeutic or diagnostic importance in large amounts, purify them and use it either as a diagnostic or therapeutic purposes. This is what is protein therapy. For example, if you are a person suffering from diabetes, he needs an insulin. So you take the insulin gene, put it either in an E. coli system or an yeast system or a mammalian system, express it in large amounts, purify insulin and give it for treatment of diabetes. This is what is being called as a protein therapy. Similarly, if a person has dwarfism because of a defective growth hormone gene, you again produce growth hormone in large amount in a expression system, either E. coli or a eukaryotic system, and then purify this and then give it as a therapeutic. Similarly, if a person is suffering from hemophilia, you can take the factor gene, express an appropriate expression system, purify this protein and give it as a therapeutic, and so on and so forth. For anemia, you can express erythropoietin and so on and so forth. This is a typical protein therapy. In fact, it has been very, very successful, and a number of therapeutic proteins made using recombinant DNA technology, using a number of heterologous expression systems are today available in the market. But the question now we are asking is that, instead of making these proteins in either E. coli or yeast or insect or mammalian cells, why can't we now take this gene and directly introduce this gene into the human cells or tissues so that the protein is made right inside our body and we can be benefited. That is the difference between a protein therapy versus gene therapy. The first human gene therapy was actually performed in September 14, 1990. So, it is about 2 decades old. The human gene therapy is about 2 decades old. This was actually done on a girl called Ashanti DeSilva. She was a 4-year-old girl. She was suffering from a genetic disorder called SCID, means severe combined immunodeficiency. Now, what is this disease? She was actually born with a crippled immune system. Now, we all have normal immune system, therefore we can fight any disease, whether it is viral or bacterial or parasitic. Our immune system can fight and we can get cured. We can protect ourselves from a number of pathogens. But in the case of Ashanti DeSilva, there was a problem in her immune system, and therefore the immune system is not well developed. That is because her body, there was a defect in a gene coding for an enzyme called adenosine deaminase, abbreviated as ADA, ADA. Now, what is the function of ADA? ADA irreversibly deaminates adenosine to inosine. This is what is adenosine, and what it does is it removes this amino group. So, deamination of adenosine results in the formation of inosine and is a very, very important step in purine metabolism. Now, what happens if this enzyme is not there? If the enzyme is not there, adenosine deoxyadenosine accumulate in the cells of these patients, and as a result, especially in the case of lymphoid cells, immature lymphoid cells, especially T cells, they are very sensitive to these toxic levels of deoxyadenosine and adenosine, therefore they die. So, these individuals will not have functional T cells. As you know, T cells and B cells are very, very important part components of immune system, and if you do not have T cells, you will not have cell mediated immune responses or even T helper responses, and as a result you cannot attack pathogens, you cannot defend pathogens. Therefore, without T cells, these ADA deficient children are wide open to attacks by viruses and bacteria, and these children therefore have the severe combined immunodeficiency disorders and is also known as a bubble boy disease, because these people all has to be in a sterile atmosphere, because they are easily susceptible for a number of diseases. For example, many common diseases that you see in the case of the SCID patients is they have 8 new ear infections per year, they have serious sinus infections, pneumonias. 2 months they have to be on antibiotics all the time, because they are highly they cannot fight many common cold and common bacteria which we normally fight with our immune system. They cannot do it, so they have to depend on antibiotics. They have a very low lymphocyte counts, less than 200. This is clinically called as lymphopenia. They fail to gain weight, they do not grow very well, and they have a lot of deep skin and organ abscesses. So, a number of problems because of this one enzyme is not made in our cell, in our body, you see the problems. The standard treatment in the 1990s for this kind of a SCID patients is they have to frequently take injections of adenosine deaminase conjugated with another molecule called polyethylene glycol called PEG-ADA. This ADA enzyme can be either purified from animals or it can be from a recombinant source, but whatever it is, the ADA enzyme has to be conjugated with polyethylene glycol and they have to frequently take this PEG-ADA so that this enzyme now can supplement the defect. So, they have to take, the problem is that as they keep taking these PEG-ADA injections, after a period of time they stop responding to this PEG-ADA, because that means they develop resistance. The PEG-ADA does not work as well as it worked in the initial periods. Therefore, by the age of 4, this small little girl called Ashanti DeSilva was no longer responding to these PEG-ADA injections. So, her T cell counts were dropping very badly. She was becoming very susceptible to a number of infections. So, she has to be confined to home and has to be taken care. The extraordinary care has to be given to make sure that she does not catch any infections. The only available alternative at that time is to do what is called as a bone marrow transplantation, but at that time, because of non-availability of matching donor, they could not do a bone marrow transplantation. So, we all are normal because we do not bother about many of these common pathogens because our immune system takes care of it. But if a person has a weak immune system, you can get infected very easily. Even a touch by someone else, or if you share a glass or even share air with air, all of them are filled with pathogens and you can catch infections very easily. So, Ashanti was largely confined to her home. So, they were desperately looking, her parents were desperately looking to see how we can keep her alive, what kind of a treatment can be given to her. So, around this time we are talking about 1990s. Around this time, a genetist called French Anderson was actually approaching the National Institute of Health to get permission to do what are called as human clinical trials, wherein he wants to introduce genes into humans in the aim of correcting genetic defects. He was trying to get permission to treat patients suffering from genetic defects by introducing normal genes. To do a human trial, especially a new form of, because nobody has done this before, people have been transfecting cell lines with all kinds of things, and people are actually testing these kinds of viral nanomolecules, animal models, and show that these things work, but to inject this kind of a viral vectors or a non-viral vector direct into humans was not heard of. So, a very strict and very complicated regulatory procedure has to be followed before he was given permission to do this kind of a methodologies in human beings. So, starting from June of 1998, Anderson's proposal was reviewed by what is called as a National Institute of Health and the watchdog in the US called as US Food and Drug Administration, and after a series of about the consultations and reviews and meetings over a period of 7 months, including 24 hours of public hearings to assess his proposal, finally he was given permission in the early 1990s to do a human clinical trial to treat patients suffering from genetic disorders, especially the severe combined immunodeficiency syndrome. So, Ashanti was the right candidate because the parents were actually looking for some kind of a permanent cure. So, Ashanti and her parents flew to NIH Clinical Center in Bethesda, Maryland, United States, and over a period of 12 days, Anderson and his colleagues Michael Bliss and Kenneth Culver isolated Ashanti's T cells and transfected them with a retrovirus expressing the ADA gene, and these genetically modified cells were introduced back into her body in the afternoon of September 14, 1990. So, September 14, 1990 was a very important landmark in the history of human gene therapy, because that is the time genetically modified cells were introduced back into a human patient by Anderson and his colleagues. The first example of a human gene therapy was conducted. So, we all have studied about the retroviral mediated gene transfer, adenoviral mediated gene transfer, and so on and so forth in the last class. So, basically what you do, you take the gene, put it in a retroviral vector, and take this retroviral vector and put in a packaging cell lines, and the packaging cells now add the viral proteins, and the replication defective recombinant retrovirus now will emerge out of these packaging cells. These recombinant retroviruses carrying the normal ADA gene is now used to transfect the cells of the patient, and then these cells which now contains a stable integrated retrovirus, and from the retroviral genome the RNA will be made and the protein will be expressed, and this protein will now be normal and the patient should be normal. So, these kinds of engineered cells is now put back into the body of the patient. This is basically the process that was done. So, her T cells were placed in tissue culture, they were stimulated to proliferate by using cytokines like IL-12 or interleukin 12. So, once the T cells are proliferating, they were infected with retroviral vector. Remember, retroviruses will only infect dividing cells. Therefore, you have to stimulate proliferation of the T cells using lymphokines or cytokines like interleukin 12, and once the T cells start proliferating, you add a replication defective recombinant retrovirus expressing adenosine deaminase. Now, the retrovirus will go and integrate into the genome of these dividing cells and the gene will be expressed, and now once we take these cells which are now transfected with the retrovirus and then put them back into her body. This was what was done for Ashanti DeSilva. This is Dr. French Anderson, and this is the 4-year little girl who was the first human candidate for undergoing gene therapy. The impact of gene therapy on Ashanti was welcomed with a lot of surprise and a lot of anticipation. Within 6 months, her T cell counts rose to normal levels. Of course, she was also receiving the normal supplementation of ADA PEG because nobody could deny the normal injections, because nobody knew how effective her gene therapy would be. But over the next 2 years, her health continued to improve, allowing her to enroll in school, venture out of the house and lead a fairly normal childhood. So, this is the picture that was taken in 1990 when she was performed gene therapy when she was a 4-year-old. This is a picture taken in 2003, and you can see about 13-year-old girl now looks normal. So, this was the first successful example of a human gene therapy performed. So, following this successful human gene therapy using Ashanti DeSilva, hundreds of labs began experimenting with gene therapy as a technique to cure disease, and more than 500 human trials involving more 4,000 patients have been launched over the last 20 years. Researchers have shown that it may be possible to use gene therapy not only to cure genetic disorders like hemophilia, SCID and so on and so forth. They can also be used for the treatment of a number of acquired disorders like diabetes, sickle cell anemia, cancer, Huntington disease, and even acquired disorders like coronary artery diseases where you have blocked arteries. They can be reopened using by expressing appropriate gene therapy techniques. So, the strategy has been, once a therapeutic gene that you have to introduce is known, you are clear about what kind of gene you have to introduce or what kind of protein has to be made, you take the gene, clone it into a viral or a non-viral vector and then introduce into the body. This has been the basic strategy for doing all these gene therapy clinical trials. Now, the type of gene therapy that was performed on Ashanti DeSilva is known as ex vivo gene therapy. That is, what you have basically done is, you have taken the cells from the patient's body, cultured them in vitro, transfected them with your gene of your interest, either using viral or a non-viral vector, and then these cells which have now contain your transgene of interest, now and you take them out and then put them back into your body. So, this is called as a ex vivo gene therapy, where cells are altered outside the patient's body and then returned. Now, there is also a more direct method, what are called as in situ gene therapy and in vivo gene therapy. In the in situ gene therapy, a healthy gene encoded by a vector is injected at the isolated site in a body that is easily accessible. In situ means you are confined to a very small region. Suppose, for example, you have developed a tumor. You can actually take a virus encoding a cytokine gene or virus encoding the alloantigens and inject this vector directly into a tumor, so that you are only injecting the gene or the vector a very localized region inside the body. So, this is known as the in situ gene therapy. A much more general approach or common method is what is called in vivo gene therapy, where the vector and gene is usually introduced systemically into the blood, so that the vector goes and then infects a number of organs, or if there are some specific receptors, it goes to targets a specific organ. It is a much more generalized and more direct form of gene therapy, where the vector and gene are directly introduced bodily, usually using a systemic approach. Okay. So, these are the three types of gene therapies usually performed: ex vivo gene therapy, in situ gene therapy and in vivo gene therapy. There are also two major distinctions. The gene therapy again that was performed on Ashanti is what is known as a somatic gene therapy, where you have actually taken the blood cells or the white blood cells or the lymphocytes from her body, introduce your gene using retroviral vectors and put them back. So, as long as you introduce your genes or the vectors into the somatic cells of body, it is called as the somatic gene therapy, where it involves alteration of the DNA of the somatic cells implicated in the disease. So, in such cases the changes that you have introduced are not heritable. So, the retroviral mediated gene transfer run can only cure the patient who has received. The children born to them will still have the same genetic disorder. So, only the person is cured because you have introduced gene only to somatic cells. The the germ cells still carry the defective gene, therefore their offsprings are still susceptible to the genetic disease. A much more ideal form of a gene therapy would be to what is called as a germline gene therapy, where you correct the defective gene or introduce your normal gene into the germ cells of the body, so that not only you are cured, but your offspring are also cured. But since the gene therapy technique that we have developed still are not very efficient, and there are also many ethical issues that are involved, so far no germline gene therapy trials have been permitted or have undergone anywhere in the world. So, germline gene therapy involves alteration of the DNA of a gamete or a fertilized egg. Changes are heritable, passed on from treated individual to the offspring, but currently no germline gene therapy is being done in humans. So, all the gene therapy that we are going to talk about today are confined to the somatic cells of humans. So, somatic gene therapy is what is going to be focus of today's discussion. There are also two different kinds of genetic disorders when we talk about gene therapy. There are genetic disorders which are caused by single gene defects. There are also genetic disorders which are called as multiple genes. So, these are called as monogenic versus polygenic diseases. So, in the case of a monogenic disease, a single locus is defective and therefore is responsible for the disease. Some of the examples are sickle cell anemia. For example, the beta globin gene is defective in sickle cell anemia. Hypercholesterolemia, where they have very high levels of cholesterol because they do not have a gene coding for the low density lipoprotein receptor. LDL receptor is not present in this gene, therefore the LDL cannot be effectively taken up by the liver cells, therefore the LDL levels become very high in the circulating blood, therefore they develop atherosclerosis and all kinds of problems. Cystic fibrosis, again it is a monogenic disease because the gene involved is called CFTR, a cystic fibrosis transmembrane regulator. Hemophilia, the defective gene is either factor 8 or factor 9, which are responsible for blood clotting. SCID, we just now discussed, the gene involved is adenosine deaminase. So, these are all examples of what are called as monogenic disorders, where the disease is because of a defect in a single gene. But there are also very complicated disorders which are called as polygenic traits. They depend upon multiple environmental factors as well as lifestyle. They are much more complicated diseases like heart disease, cancer, diabetes, alcoholism, schizophrenia. These are all called as polygenic traits, and it is not because of a single gene that you get into these disorders. Complex interactions between the genetic complement and the environment and as well as your lifestyle can lead to these kinds of disorders, and we are still not equipped to treat any of these polygenic disorders. So, all the discussion about human gene therapy is being focused on somatic gene therapy as well as monogenic disorders. These are the two major areas of gene therapy that is currently going on in the case of the human gene therapy. So, some of the examples I have given here, what kind of monogenic disorders are being treated or or being aimed to be cured by gene therapy. We still have not cured any, there are still in clinical trials, are being aimed to be cured by gene therapy. Things like SCID ADA deficiency, alpha-1 antitrypsin deficiency, this leads to a disease called emphysema, chronic granulomatous disease, cystic fibrosis, familial hypercholesterolemia, Fanconi anemia, Gaucher's disease, Hunter syndrome, Parkinson's, hemophilia. These are some of the diseases which are all examples of monogenetic disorders where the defect is in a single gene. Therefore, if you can introduce the correct gene by a viral or a non-viral vector, can you cure this disease? This is what is being aimed at. So, I will just give you 2 or 3 examples of gene therapy for genetic disorders just to get an idea. There are a number of such disorders. At the end of this lecture, I have given a series of references. One can always go through these references to get yourself updated on various other kinds of genetic disorders. Hemophilia is one of the very attractive candidates for gene therapy. The hemophilia is an X-linked disorder affecting 1 in 10,000 males. The disease is actually due to a defect in a gene coding for a blood clotting factor called factor 8. There is also what is called the hemophilia B, where the defect is because of a defect in the gene coding for factor 9, another clotting factor. Normally, in all the normal individuals, the circulating levels of factor 8 is about 200 nanograms per ml. The factor 8 is produced from the liver and is secreted into the blood. So, all of us are normal, because whenever we have a cut, the blood immediately clots. That is because the factor 8 is, we are all producing factor 8, and we have about 200 nanogram per ml in our blood of the factor 8 protein. But even if you maintain 0.1 to 0.2 units, that is 1/10 or 1/20th of these levels, you can still, your blood can still clot, and it is still considered clinically significant. So, what is now being aimed at in the gene therapy for hemophilia is, can you use a non-viral vector or a viral vector to introduce the gene coding for factor 8 into these hemophilia individuals, so that they can maintain about 0.1 to 0.2 units, which is 1/10 or 1/20 the normal levels of factor 8. Unfortunately, even this we have not been able to successfully achieve till date, because there are a number of problems associated with gene therapy still. So, the strategy has been introduce the gene encoding factor 8 into patients suffering from hemophilia using viral or non-viral vectors, such that clinically significant levels of factor 8 can be produced in their bodies. This is what is being aimed for hemophilia for gene therapy. Similarly, if you take a disease like familial hypercholesterolemia, it is a genetic disorder characterized by high cholesterol levels, especially very high levels of low density lipoprotein, which is called as the bad cholesterol, in the blood as well as in the blood, leading to early cardiovascular diseases. Many patients suffering from familial hypercholesterolemia have mutations in the LDLR gene, which codes for the low density lipoprotein receptor protein, which normally removes LDL from the circulation. They may also have mutation in another gene called as apolipoprotein B, which is part of the LDL that binds with the receptor. So, when you have mutations in either of these two genes, it can manifest in the form of a familial hypercholesterolemia. They have very high levels of cholesterol in the blood, because the LDL is not taken inside the liver, because the liver cells do not express LDL receptor gene. So, what is normally done in such cases? What is being aimed to see, can you now target using a non-viral or a viral vector, can you target an LDL receptor gene into the liver cells of these patients, so that liver cells now express LDL receptor, therefore, LDL can be taken inside the liver and the levels of LDL in the blood can be lowered. This is what is being aimed at. Perform either ex vivo or in vivo gene therapy using viral or non-viral vectors encoding the LDL receptor genes. So, take hepatocytes from these individuals, culture these hepatocytes in vitro, allow them to proliferate, transfer them either retroviral vectors or adenoviral vectors or non-viral vectors, and once the gene got transfected, take these hepatocytes back and infuse them back through the portal vein into these animals. So, liver cells go and repopulate the liver, and now the liver start expressing LDL receptor gene, and the LDL can be now cleared from the circulation. Or using again non-viral or viral vectors, directly introduce the gene into the liver, so that the using in vivo gene therapy, so that the liver cells now starts expressing LDL receptor. This is what is being aimed at. Similarly, another important disease called ornithine transcarbamylase deficiency, OTC. Now, deficiency of ornithine transcarbamylase, OTC, is inherited as an X-linked recessive mutation. The job of OTC is, it normally breaks down amino acids present in the dietary protein. If you do not have OTC, it leads to buildup of ammonia, which damages brain function. So, they go into coma. Low protein diets and ammonia binding drugs are used to treat OTC deficiency. Clinical trials to treat OTC deficiency were established using adenovirus as a vector for the normal OTC gene. So, you now take the OTC gene, put it in adenoviral vector, and see, can you take these recombinant adenoviruses and directly inject them into these OTC patients, so that they now start expressing the enzyme and patients can be cured of this disease. These are a couple of examples of genetic disorders being aimed for cured by gene therapy. A number of acquired disorders are also targets for gene therapy. For example, gene therapy for the treatment of cardiovascular disorders, revascularization of blocked coronary arteries leading to myocardial infarction. We know the coronary arteries, when they get blocked, it results in the myocardial infarction or heart attacks. So, what is being aimed by gene therapy is that, can you introduce vectors, non-viral or viral vectors encoding angiogenic factors like vascular endothelial growth factor or fibroblast growth factor, VEGF or FGF, that promote growth of new arteries leading to revascularization of blocked arteries at the site of ischemia. So, new blood vessels can grow around the infarct, therefore, the heart will now get new blood supply. Can we now aim at it? So, can you now introduce vectors, either adenoviral or retroviral vectors, to the site of this ischemia and promote the growth of new blood vessels by expressing genes encoding vascular endothelial growth factors or fibroblast growth factor. This is what is being aimed for, gene therapy for treatment of cardiovascular disorders. Similarly, local expression of antithrombotic genes. For example, gene therapy aims at dissolving blood clots following angioplasty by introducing vectors encoding cyclooxygenase 1, which is involved in synthesis of prostacyclin, which is an antithrombotic agent. So, these are some of the examples of treating acquired disorders. These are not genetic, these are acquired disorders using gene therapy. Again, gene therapy for the treatment of cancer. Again, one of the early successes or the early human trials of gene therapy were performed in the area of cancer, wherein treatment of metastatic melanoma by adaptive immunotherapy using what is called as tumor infiltrating lymphocytes with vectors encoding interleukin 2. Now, we have our immune system, and normally the lymphocyte should recognize these tumor cells, because these tumor cells express many foreign proteins or very high levels of proteins which are not normally expressed in the normal cells. Therefore, these lymphocytes somehow should have recognized this tumor as foreign and destroyed. But for some reason, these lymphocytes are unable to recognize these tumors as foreign, although there are lymphocytes in the vicinity of the tumor cells. Therefore, they are called as tumor infiltrating lymphocytes. So, what you do is you isolate these tumor infiltrating lymphocytes and then introduce by viral or non-viral vectors genes encoding cytokines like interleukin 2, and these tumor lymphocytes expressing high levels of interleukin 2, if you now inject them back into the body, they now go and then go to the tumor and now express very high levels of interleukin 2, and therefore attract T cells to the vicinity of tumor, and now the T cells will go and then recognize the antigens being expressed on tumor cells and will destroy the tumor. This is one approach. Another approach is to use viral or non-viral vectors including tumor suppressor genes, cytokines, alloantigens, and so on and so on. In fact, more than half of the clinical trials, or even more than that, of the human trials that are going on in the area of gene therapy is aimed at treatment of various type of cancers. So, a large number of human trials are going on aimed at treatment of cancer using gene therapy. Gene therapy for arthritis, delivering anti-arthritic genes intra-articularly into the individual joint, where their expression leads to accumulation of sustained therapeutic levels of the gene product. Now, what kind of genes you are trying to introduce for treatment of arthritis? I have given a list here. You can introduce, for example, IL-1 receptor antagonist, HSP thymidine kinase, tumor necrosis factor receptor, FC fusion, commercially known as etanercept, using vectors like adeno-associated virus. Here, people are trying to introduce these genes using retrovirus vectors, transforming growth factor beta, and so on and so forth. So, using either retroviruses or direct plasmid non-viral vectors involving plasmid DNA or adeno-associated virus as vectors, people are trying to introduce a number of anti-arthritic genes directly into the joints to see localized expression of these anti-arthritic genes, can they have a therapeutic effect leading to the cure of arthritis. Now, in all these cases, there are many such examples. I will not go into all the details. One can always go and look up into all kinds of diseases that are being aimed for gene therapies. The three major challenges in all these, whether it is for a genetic disorder or acquired disorder, the three major challenges in gene therapy are delivery of the genes, sustained expression of the delivered gene, and regulation. These are the three major issues for gene therapy. Whether it is a genetic disorder or acquired disorder, the major issue that being addressed is, can you now deliver the gene to as many cells in the body as possible? Efficiency of delivery is very important. And once you deliver the gene, can the delivered gene explain in a sustained manner? The major problem that you have with all the viral and non-viral vectors that we have discussed in the last few classes that are being used for gene therapy is that they all express, because the whole idea of gene therapy is to see, for the rest of his life he does not require protein therapy. That is what is the ultimate aim of gene therapy. That means, once you perform a gene therapy on an individual, the protein should be made for his lifetime. Even we have not, so far we have not achieved that kind of a results, but even the introduced gene can express us for, let us say, a few months or a few years, it is a major breakthrough, because protein therapy you have to take at a much frequent intervals. So, the introduced gene should be able to express for sustained periods so that you have a long term advantage. But for one of the major problems in gene therapy is that whenever you introduce these genes using either retroviral vectors or non-viral vectors or other adenoviral vectors and so on and so forth, the expression is there for initially a few weeks or few months, and after some time the expression drops down. And they say this is one of the major problems. Lack of sustained expression is one of the major problems why gene therapy has not been still a clinical reality. Again, regulation. Many of the vectors that we are using use constitutive promoters like the cytomegalovirus promoters and so on and so forth. So, the introduced gene is constitutively expressed, but ideally you would like to express the introduced gene also to be regulated the same way as the endogenous gene. It should have its own native promoter and so on and so forth. So, the regulated expression of the introduced gene is another dream of gene therapists all over the world, which we still have not achieved. So, the major steps in performing gene therapy is that you need to package the gene, that means you need to put in the right vector system, either viral or non-viral. You have to protect the gene, you have to make sure that when you introduce this gene into the body, it should not get destroyed either by the immune system or by nucleases and so on and so forth if it is a non-viral vector. And then you deliver the gene to the nucleus and express it over a sustained period of time. These are the three major objectives of gene therapy. So, to achieve this, what is the most important criteria or what is the most important step in performing gene therapy is to develop what are called as safe and efficient vectors. Whether it is viral or non-viral, you need to have safe and efficient vectors to achieve all these three major challenges that are required for successful gene therapy. So, what is an ideal vector for gene therapy? An ideal vector would be it should be able to carry one or more genes of your choice. It should be targeted, that means for example, if you want to treat familial hypercholesterolemia, your gene should be expressed only in the liver, not in other tissues. So, your vector system should be designed in such a way that it will only target to the liver. Similarly, if you want to treat a disease like cystic fibrosis, your vector system should be in such a way that it will only go and target to lung tissue. But there are also diseases like hemophilia where your objective is to express factor 8 or factor 9 in the blood. So, in such case it does not matter, you do not require a targeted vector, does not matter whether the vector is expressing your gene in muscle or liver or lung, as long as the factor 8 is released in the blood and you are able to maintain clinically significant levels of factor 8, you have achieved your objective. So, there are certain genetic disorders where targeting is not required, but there are certain disorders where targeting is very efficient. For example, Parkinson's disease, you have to introduce the gene coding for tyrosine hydroxylase right here into the substantia nigra in the brain, only then it will have a desired effect. You cannot introduce in liver or anywhere else. Similarly, if you say arthritis, you have to target the gene or vector into the joints, only then you will have a clinically beneficial effect. So, targeted delivery and expression of the vector is very, very important. The vector should not induce any immune response. One of the major problems with some of the vector systems, both non-viral as well as especially adenoviral vector systems, is that when you introduce, suppose the expression level goes down after say 3 or 4 months, when you again re-inject the vector, some of the vector encoded proteins which are present on the vector, they would have induced an immune response in their first time. So, when introduced the second time, our immune system will recognize it as a foreign and the vector will be destroyed. So, even before the vector gets into the target tissue, the vector will be eliminated by your immune system. So, one of the major objectives of gene therapy is to develop vectors which are not immunogenic. So, the vector should not elicit an immune response from our body. It should be stable, it should not undergo any mutations, it should be easy to produce, especially if you are talking about viral vectors, you should be able to produce high concentrations of these viruses using packaging or producer cell lines, because cost is one of the major factors in gene therapy. Regulatable, as I said, you should produce enough protein to make a difference, and ideally should also be regulated the same way as the endogenous gene. So, these are some of the major characteristics of ideal vectors. Unfortunately, either the non-viral or the viral vector system that we discussed in the last 2 classes, none of them actually fulfill all the criterions of an ideal vector. So, we do not have a vector system which fulfills all the criterion of a ideal vector. So, there are still problems. So, despite all these deficiencies and drawbacks, a number of human gene therapy trials are going on across the world. In fact, this is a data I took out from one of the journals published in 2007, and you can see the number of trials that are going on across the world using various vector systems: adenovirus, retrovirus, vaccinia, pox viruses, non-viral, and so on and so forth. A number of trials are going on, and as you can see, majority of the trials involve the use of either adenoviral vectors or retroviral vectors and non-viral vectors. So, these are the three most popular vectors that are being used for a number of gene therapy trials, namely the adenoviral vectors, retroviral vectors, and non-viral vectors. Now, so having discussed what is the general strategy for gene therapy and what kind of diseases are being cured at, and how to introduce the gene, whether it is in vivo or in situ or ex vivo, and so on and so forth. As I said, the first human gene therapy trial was done in 1990, so it is now 2 decades. What has happened now? Have we really achieved the ultimate objective? Has human gene therapy has become clinical reality? The human gene therapy is still not a clinical reality because they are still under various stages of clinical trials. Still, it has not reached a stage where you can go to a clinic and then ask the doctor to perform gene therapy for either a genetic or an acquired disorder. Why is the reason? What is the problem? What are the problems associated with all these 2 decades of human gene therapy research? Let us now look at 2 or 3 of such major problems which hindered or which hampered gene therapy becoming a clinical reality. I listed about 4 major issues which were the major challenges and major problems and major failures of human gene therapy. In the year 1999, when a adenoviral vector was actually used to treat a human patient to cure ornithine transcarbamylase deficiency, which we discussed just a few slides back, this patient actually died when he was undergoing gene therapy. Similarly, in the year 2002, in France, some human patients who were actually treated by gene therapy for SCID, severe combined immunodeficiency syndrome, some of them developed cancer, leukemia, where the retrovirus was actually used for transferring your gene. Similarly, in 2006, a woman who was treated for X-linked chronic granulomatous disease using retroviral vector also died. And in 2007, a patient who received a rheumatoid arthritis underwent treatment for rheumatoid arthritis using adeno-associated virus controlled anti-arthritic gene also died at the time of during the gene therapy trial. So, from 1999 to 2000, there have been incidents of patients who received the gene therapy treatments, if not all or some of them actually died or developed a lot of problems. So, I will just spend some time to discuss what these problems are. For example, in 1999, this gentleman known as Jesse Gelsinger, who was suffering from ornithine transcarbamylase deficiency, was taken in for a human gene therapy trial, wherein adenoviruses expressing the OTC gene was introduced into the body. The gene therapy began on September 13, 1999, but Gelsinger went into coma on September 14 and he never recovered. He died. And the cause was identified as respiratory distress syndrome. Now, a lot of investigations went on to see why did he die, what was the problem, and it turns out it is the problem is the vector. Adenoviruses, when you use in very high titers, can cause a problem. So, adenovirus vectors, it was decided that adenovirus vectors are better used for killing cells, especially for cancer gene therapy, than for a genetic disorder, and dose escalation studies should be better controlled. You cannot inject very high doses of these adenoviral vectors, and you need to use adenoviral vectors which are non-immunogenic, which are less immunogenic. These were the conclusions. In fact, the death of Jesse Gelsinger in gene therapy trial in the year 1999 was one of the most highly investigated gene therapy trial failures, and a number of papers have been published to understand what exactly was the cause for the death. It is published in very respected journals. In fact, there is a very nice article written by the editor of Human Gene Therapy, which discusses the background events that led to the death of the Jesse Gelsinger in September 1999. So, once this kind of a problem went in, a lot of people went in to understand what was the problem and why this failure happened, so that these kinds of trials were briefly stopped, but once conclusions are drawn, you make sure that you do not repeat some of these mistakes and you resume the gene therapy trials. The other important drawback that happened was in Europe, especially in France, when about 12 to 14 patients were actually treated for SCID, severe combined immunodeficiency syndrome, again using retroviruses for gene transfer. The successful treatment of these patients was greeted with excitement and was published in Science, a very reputed journal, in the year 2000-2002, saying that they had a clinically benefited results. And you can see here 3 to 5 years after performing the gene therapy for SCID, of 14 out of 15 children were able to live normal lives instead of living in the sterile bubble. They had normal numbers of T cells of both CD4 and CD8 subsets. They responded to childhood immunizations. That means the immune system is, they had very good T cells. The T cells are now able to respond to vaccinations, including diphtheria, tetanus and polio, by producing T cells and antibody specific for these antigens, indicating that the immune system became normal. The antibody production is sufficiently good that they have no need for periodic infusions of immunoglobulin. So clinically it was very successful because they had very clinically beneficial results following the gene therapy. But the problem was 2 of the 10 children treated in France developed leukemia like conditions. So the optimism that was generated by what was considered to be a first true success of gene therapy turned into disappointment at the news that 2 out of 10 children actually developed leukemia. In fact, genetic analysis of the malignant cells actually showed that when they used the retroviral vector for introducing this gene for treating the SCID, it turned out this retroviral vector went and integrated into this very specific region in the human chromosome, and as a result it activated an oncogene called LMO2. So when the retrovirus went and integrated here, because these retroviruses are very powerful LTRs, it activated this oncogene, and when these oncogenes were overexpressed, it led to leukemia. So this the culprit turned out to be integration of the retroviral vector to a very specific region, resulting in the activation of oncogene was responsible for development of leukemia in these children who underwent gene therapy. So the activated gene was not only the cause of malignancy, but was also the likely event that triggered leukemia. So again, all these were investigated thoroughly. So what is very important about gene therapy is that once they identified the problem, they went on and investigated and identified what is the cause. Why did these children got leukemia, and they found out this is the molecular mechanism by which these children develop cancer, and therefore attempts are now being made to see how we can avoid and how we can in the future trials. And in fact, a few years later, another child, the third child also developed cancer, who underwent the same thing. So the first trial was stopped in October 2002, and other trials were started 3 months later after 2 children in the trials developed cancer. But on 24 January 2005, the French Medical and Regulatory Authority announced that a child who was treated by the same Fisher group in April 2002 also now has cancer. So 3 out of 10 children developed leukemia or cancer, indicating that there are still problems with retrovirus mediated gene transfers. So some more research needs to be done. Similarly, in the year 2007, a woman who was undergoing treatments for arthritis, she was actually being treated in July 2007 using a recombinant adeno-associated virus expressing a fusion protein consisting of extracellular domain of human tumor necrosis factor type 2 and Fc domain of IgG1, was injected to a 36-year-old woman who was suffering from rheumatoid arthritis locally into the symptomatic joints, with the expectation that the protein which is produced from this adeno-associated viral vector will be produced intra-articularly and will have a local therapeutic effect. But unfortunately, 22 days after receiving the second dose, this woman died. And early in 2007, also again this case was investigated, and soon it was realized that the cause was not really gene therapy, and in fact Food and Drug Administration removed the halt of the clinical trials and allowed the study to resume with minor changes in the protocol. Now, what all these failures actually told is that people went and identified what is the molecular reason, what is the cause for this particular problem, and they tried to rectify it, and again the future trials these problems were rectified. Now, what are the positive developments? So I showed you so far that yes, of 1 or 2 very successful human trials, some of them died and some of them developed cancer, and as a result there is only a cautious optimism as far as human gene therapy is concerned, and these are some of the reasons why human gene therapy, despite 2 decades of research, has still not become a clinical reality, because we still have not perfected the procedure. But what are the positive developments? The first world's anti-cancer gene therapy drug is out in the market. This is known as Gendicin. It is actually a recombinant human adenovirus expressing P53, which is a tumor suppressor protein, and it was injected into patients suffering from head and neck squamous cell carcinoma. This was done in China, headed by Professor Zhang Xuanwen of Beijing Cancer Hospital. They completed all the clinical trials, and finally the Chinese regulatory authority granted license on October 16, 2003, and this is the world's first anti-cancer gene therapy drug. In fact, there is a very nice article in Human Gene Therapy, a very prestigious journal in this area, about the commercialization of Gendicin. Gendicin, the first commercial gene therapy product. So there have been some positive developments too in the area of human gene therapy. Again, gene therapy for Parkinson's disease. Now, what is Parkinson's disease? In these patients, patients lose dopamine producing brain cells, resulting in substantial reduction in the activity and amount of a neurotransmitter called gamma aminobutyric acid, a major inhibitor neurotransmitter in brain. This controls the locomotory organs of the body. As a result, it contributes an abnormal increase in the activity of a region in the brain called the subthalamic nucleus and a key regulatory segment of movement, and causes a dysfunction in the brain circuitry response for coordinating movement. That is why you see these patients, they cannot coordinate their movements anymore. A company called Neurologics Incorporated is now trying to see, can you cure this Parkinson's disease by gene therapy using adeno associated viruses expressing the gene of the interest. And in fact, this particular thing, if you go to their website, it tells you that they are now in phase 2 clinical trials, and just this year, just in June 2010, the company announces very successful phase 2 trial of gene therapy for Parkinson's diseases. So the study participants who received this particular AAV vector encoding the gene of interest for Parkinson's disease experienced statistically significant clinically meaningful improvements in off medication motor scores compared to control subjects who received sham surgery. The results also demonstrated a positive safety profile, that means there were no serious adverse effects and it had a clinically beneficial signs. So these are all very positive developments saying that yes, at least for certain diseases, gene therapy may become a reality very soon. Just today is actually September 23rd, 2010. Last week in Nature, a very again exciting development took place where transfusion independence and HMGA2 activation after gene therapy of human beta thalassemia. So just last week a paper was published in Nature clearly showing that beta thalassemia was successfully cured by gene therapy, and tells you that in this particular case the gene therapy actually began in the year 2007. So after 3 years of this trial, and what they have actually shown, in this case they have actually used the lentiviral vector for introducing the beta globin gene into these individuals, and this patient who was actually dependent on monthly transfusions of beta globin gene, now 21 months after gene therapy he has not received any transfusion and is all right, and blood hemoglobin was maintained between 9 to 10 grams per deciliter, of which 1/3 contained vector encoded beta globin. That means for a period of 21 months the vector which was the lentiviral vector which was expressed in the beta globin gene is successfully expressing the gene, and he is not required any blood transfusion so far. So these are very positive developments indicating that yes, in this century there is a good hope that gene therapy will become a clinical reality. There are also a number of gene therapy trials going on for correcting a number of retinal disorders where you use viral or non-viral vector directly inject into the eye, and these are called as ocular gene therapy. I will not go into the details because of lack of time, and there is a very recent review in the last month's issue of Human Gene Therapy on ocular gene therapy, an evaluation of recombinant adeno-associated virus mediated gene therapy for treatment of a number of ocular diseases. One can go and look up these things. So what are the conclusions from all that I have said so far? It would be unrealistic not to expect gene therapies to produce side effects. There will be side effects, there may be problems, but gene therapy continues to require informed use in controlled clinical studies with clear consideration of the risks and potential benefits. Current vector systems may need to be modified, and additional efforts are required to better understand the biology of the diseases which are candidates for gene therapy. Together, this information will enable risk classifications for specific vectors and transgenes, as well as assessment of risk factors that are unique for each clinical trial. And with this approach, the therapeutic potential of somatic gene transfer may be realized through the application of appropriate preventive strategies. So, we are hopeful that we will learn from all these mistakes, and with some of the success I have told, gene therapy will soon become a clinical reality. There are a number of resources for understanding gene therapy and then gaining more information. In the next few slides, I have just listed some of these resources. There is what is called as a National Gene Vector Laboratory in the United States, which actually distribute a number of vectors which are being used for gene therapy. One can just write to them and get a number of vectors. They even make some of these vectors and use it in clinical trials. There is also what is called some National Gene Vector Biorepository located in Indiana University of Skidison, which again assists gene therapy researchers across the globe. There are very specialized journals which are required for, which are now available for publishing gene therapy trials and gene therapy results and gene therapy research. There is a journal called Gene Therapy published by Nature Group. A number of review articles are there. If you just go to the website, many of these review articles can be downloaded free. There is another very important journal called Human Gene Therapy, produced by Marianne Libert. Again, a number of interesting articles discuss about the progress being made. There is a very dedicated journal called Cancer Gene Therapy. A number of gene therapy trials going in the area of cancer treatment. There are beautiful review articles. This is, for example, appeared in the Annual Review of Biochemistry discuss about the various vectors that are being used for gene therapy. I have also listed a number of review articles. One can go through all these review articles and enlighten yourself about various gene therapy things. Some of the very popular reviews written by some of the leading researchers in the field are listed here. Gene therapy using non-viral vectors. This is the first report of introducing a naked DNA into skeletal muscle and demonstrating expression. This led to using naked DNA for gene therapy, published in Science in 1990, and then people started modifying this plasmid gene therapy using a number of reagents. You could improve the efficiency of gene transfer using gene gun, electroporation, or using cationic lipids. A number of such articles one can read and understand how non-viral gene vectors are being used for various purposes. For the treatment of cystic fibrosis, how cationic lipid mediated gene transfer is being used for treatment of cancer and cystic fibrosis. So, I think I will stop here. I hope that I have convinced you that human gene therapy is a very, very exciting area. There have been certain drawbacks, there have also been some very positive developments, and hopefully some of these things will become a reality, and soon gene therapy will become a clinical reality in the next couple of decades. Thank you.